All right, so welcome everybody. Thank you for attending our second Digital Health Roundtable presented by UCB's Digital Care Transformation team. The title for today's session is Creative Evidence Generation Strategies Implementing Real-World Data and Real-World Evidence. My name is Emily Lewis. I'm a Digital Business Transformation Lead at UCB on the Digital Care Transformation team. And I'm joined by 4 panelists today, each with a diverse experience within real-world data and real-world evidence. The first is Dorothy Bartels. She's the Global Head of Real-World Evidence and Digital Sciences at UCB. She is a pharmacoepidemiologist by training with 15 plus years of experience in both the pharma and payer spaces. She's also served as Clinical and Real-World Data Strategy Lead at Google's X, the Moonshot Factory. And she's also been a part of 2 professorships and has close ties to academia, and she's passionate about digital epidemiology. So welcome, Dorothy. Second panelist today is Dr. Ying Liu. He's a professor of biomedical data science at Stanford University School of Medicine, where he's also the co-director of the Center for Innovative Study Design. He's got over 30 years of experience working in academia and in government, and he's served as a consultant to the pharmaceutical industry. And the fun fact is that he's also a PI and part of our UCB Stanford collaboration, where he's leading a project integrating real-world data from registries into ALS clinical trials. Welcome, Dr. Ying Liu.
Thank you.
Our next panelist is Dr. Jeremy Rassin. He's the co-founder and president of Aetion, which is a healthcare technology company that delivers real-world evidence for life sciences companies, payers, and regulatory agencies. He is also a pharmacoepidemiologist, and he has 25 years of experience in both academia and industry, and he's focused on methods to improve the quality and validity of real-world data studies. Welcome, Jeremy.
Thank you. Thank you for having me.
Last but not least, we're joined by Devin Gilliam. He's the head of clinical trials at Datavant, where he's currently pioneering trial tokenization technology to bring more real-world data into clinical research. He's got a bachelor's degree in neuroscience and a background in healthcare consulting and business development. Welcome, Devin.
Thanks for having me.
So to start off today's conversation, I wanted to kind of set the stage about real-world data and real-world evidence and the difference between the two. And luckily, the FDA has kind of come out with some standard definitions for us to use. So starting with real-world data, it's the data relating to patient health status and the delivery of healthcare routinely collected from a variety of sources. And I have those sources, some of them, represented here on this next slide. So it could be claims and billing, omics, lab data, patient-generated health data through mobile apps and devices. Could also be data on social determinants of health and environmental data. Could be electronic health record data. It could be remote patient monitoring data. It could be patient-reported outcomes or PROs. It could be medication or pharmacy prescription data. And it can also be data on resource utilization, as well as morbidity and mortality data. So as you can see, there's a smattering of different sources and types of data that fall under the umbrella of real-world data. And since there's so many, it might be best to sort of look at bucketing those types of data into 4 categories. And you can sort of think of it as point of care clinical data, patient-generated health data, cost and utilization data, and public health data. So that helps, at least for me, to kind of think about it in sort of a segmented way. Real-world evidence, on the other hand, is defined as the clinical evidence regarding the usage and the potential benefits or risks of a medicinal product derived from the analysis of the real-world data. So examples of this are any type of research method being applied to that data. So it could be open label trials, randomized controlled trials, observational trials, and even pragmatic trials in which we have a more diverse patient population and care setting represented. So to kind of also think about transitioning into our discussion, I wanted to kind of think about the old way and sort of the new way of using real world data. And really what we saw previously was data only being used post-market, and that was used to look at study treatment patterns, monitor drug compliance and adherence, pharmacovigilance monitoring, comparative effectiveness with other competing drugs, and also looking at demonstrating the value of the drug to optimize treatment pricing. So it was really in this post-market space that real-world data was being used. In recent years, we've seen sort of this new way being defined, which is both pre-market and post-market. So we're looking at using real-world data to identify unmet patient needs, profile and segment the target patient population, optimizing the trial design, and really ultimately to complement and sometimes supplement prospective clinical trial data, and providing more of a totality of evidence approach for an evidence package for somebody like the FDA or a regulatory authority. So that's sort of where we are now, and it's a very exciting place to be. And lucky for us, we have some stats from ATN where they've dug in a little deeper to what's been happening pre-market. So they did some studies, and they looked between January 2019 and June 2021 and found that 116 out of the 136 submissions to the FDA that occurred during that time, roughly 85%, incorporated real-world evidence in any form. And of those, 88 included a real-world study intended to provide evidence of safety or effectiveness. And of those, 65 actually influenced a final approval or decision, and 38 were actually included in product labels. So this is all really encouraging. We're definitely seeing an uptick in the use of real-world data pre-market. And that's where I want to focus our discussion today. So let me stop sharing my screen and dig into the conversation with our experts here. So the first question I have for you all is, how should real-world data and real-world evidence inform hypothesis generation and testing? Does the feedback loop that real-world evidence provides allow us to ask better questions to begin with? And should those types of questions that we ask be any different? I'll open it up to you all.
Thank you. Maybe I take the opportunity to answer this question. And first of all, thank you very much for inviting me to join this discussion, and also thanks to UCB to support Stanford research, and particularly this collaboration, so that we can explore our methods. So I want to say that, hypothesis generating has traditionally based on observational studies. So it's not very surprised that we use real world data to generate more hypotheses. What makes differences here is that, first of all, the type of data that available has been expanded tremendously thanks for the technology advancement. The second part is the analytical technology that developed allow us to really look into high dimensional complex and not necessarily linear relationship among those data. So allow us to find associations that can help us to generate more global and deep hypotheses. Examples like this is for example, that VA Milling Veterans Program that allows the program was developed to get consent from veterans, milling veterans, who allowed to do their genetic tests, and then allow the link of genetic tests to their real-world electronic medical records. So this is very powerful to identify new biomarkers or genetic predictors for the disease onset, disease progress, and disease outcome, and help to identify the new pathway, a new particular pharmaceutical targets. And particularly interesting is that also allows to identify subgroups that maybe have different pathways. And for example, a large portion of VA veterans, including the not necessarily majority, but the huge number underrepresented population that developed in clinical trials. And so those targets may miss from the trial community, but really brings some new information. And our colleagues at VA and Stanford done great work there. I'm not be able to share their findings, but those findings potentially will direct the new drug targets. And secondly, so that will be really in the R&D level. And secondly, being used tremendous, as you said, post-marketing safety, right? And recently, my colleagues at Stanford, including myself, with the Danish team, used the 10 years birth cohort at Denmark. And then the unique part is that birth registry can link with a prescription of father's medication. And so we actually discover a signal of metformin usage with this birth defect among children, and published this year in the Annals of Medicine. And these kind of findings was not possible if we don't have real world data. So that feedbacks a new hypothesis that needs to be tested in more rigorous way to say if there is just observational or why that happens. And also we have been using the real world data to help design, so in the clinical pre-marketing stage, to design clinical trials. It has been known that we're using real world data to identify endpoints, to see where's the availability of patients. And but also recently, I think Genentech and the Stanford team has worked together to develop a AI tool called Pathfinding to explore the eligibility criteria, to relax the eligibility criteria for oncology trials based on Flatiron real-world data. And so basic idea is construct a counterfactual experiment and to see what are the eligibility criteria can be relaxed without impact on the trial conclusion or efficacy power. So these are very powerful tools that actually bring new insights to the problems that we otherwise would not do. And not to mention also real world data can help for the patient level that decide what will be the best treatment. So post-marketing part. So overall, I think the type of question that we can ask, maybe for the real world data, we need to expect that the data be there and accurate and available. So we're not using real world data to ask them, how the drug works, why it works, like mechanisms of biology, but it will be work much better for questions that can be answered using large number of data and also using conventional collect endpoints that fit the use for the clinical questions. So very much like, we're trying to advocate over times that in the early phase trials, you want to do very detailed scientific integration to explore the mechanisms, why it works, and inter-biology relationship. When you move to phase 3, the ideal world, you want to do a large, simple trial. And so real world data is more like even beyond the phase 3 trial, and should be focused more of the questions that can be answered using large amount of data and also some kind of simple endpoint, relevant endpoint. So that's my point. Thank you.
Yes, and I jumped just in, and also big thanks from my side. And I think the point you made is super important from my perspective as well, to look into the whole drug life cycle. And so it may change. So in the early days, real-world evidence, there might be maybe much more hypothesis iterations and quick and fast. And in the later stage, it's more targeted hypothesis generation and testing. So then it's probably more about identifying new subgroups or label extensions. So I think it's highly dependent on the drug life cycle stage. Thank you.
I'll just add an example that that I was part of in the COVID space. I think there's a real possibility in the earlier stages, and Emily, I think this goes to your diagram where you sort of move the circle to include earlier parts of the life cycle, where theories that come out of the lab can actually be tested or validated in some way in real world data. So this was an example, as I said, in the COVID space, where researchers predicted what the shape of, I'm going to get this wrong because I'm not a biologist, but essentially what the shape of a molecule would need to look like in order to interfere with the COVID replication process, and found some existing drugs that had shapes that were similar to that. We can use real world data to test that hypothesis, because as a colleague of mine used to say, or probably still says, in the real world everything happens. So these are not terribly common medications necessarily, but with enough people doing enough things, you can see some pattern of use there. So you look at patients who got COVID and happened to be on this drug versus got COVID and didn't happen to be on this drug, and you can start to compare to see if these lab-driven hypotheses can prove out in the real world. Now, obviously, the drug that exists isn't the perfect drug, but you can use that in a development program to drive the directionality of that program and to validate some of the hypotheses that underlie that program. So I think there's some really interesting possibilities for the use of real world data and real world evidence.
We're seeing some hypothesis generation that actually occurs during phase 1, phase 2, and it often is the case that obviously more than half of these drugs will actually fail, and the reasons for the drugs failing can be quite interesting, and you can start to learn about the patient population a little bit better by linking in real world data. So an example is linking historic information on these patients, perhaps getting their the previous treatment or previous diagnosis history. I think what's really compelling is starting to link more and more cancer patients, if you look at specific tumor types, are having next generation sequencing. That data is really hard to access from a clinical trialist perspective. If you could actually pull it back in and better define your population with genomic information, you start to better understand things like super responders, for instance. So rather than considering a phase 2 asset fail, you can perhaps implement and run a better designed next phase 2 trial to better target the population that most benefited from that drug.
Well, thank you for all of those excellent examples. And actually, Denvin, you paved the way to the next question, which is really about study design itself. So thank you for that segue. So yeah, the next question really is, how can we better design trials using, or randomized controlled trials using real world data and real world evidence? And first of all, like how do we determine the most relevant data elements to use? And then how do we assess the suitability of the data sources that we go to retrieve that data?
So I can start with this one. I think you mentioned in your intro, Emily, about RWD, longstanding use of it in commercial use cases. We've started more recently to see some compelling use cases around really cool stuff like external control arm development, real-time identification of patients for clinical studies. But this is still all the use of RWD kind of outside of that clinical trial environment. Where I think we're seeing a bit of a paradigm shift is, okay, how do we actually inform data collection within our clinical study by enabling sponsors, researchers to actually start to collect and integrate, exogenous or I guess we could call it real world data. So one Datavant, the company that I work for, one thing that we're pioneering is the use of tokenization or privacy preserving record linkage to actually connect these disparate real world data sets and bring them back to researchers to better understand kind of the patient populations. You could potentially use it for prospective data collection to get a better understanding of longer term outcomes and safety events in patient populations. And I think what really matters is things like data completeness and the type of data to answer the right questions. So we know, for instance, open claims data, it's limited in certain applications, better suited for others. Sometimes you need high fidelity EHR data. So it's just really defining that ahead of time of where can we find the data? What does that data look like? And how representative is it of the patients that are actually enrolled to our study? And then is it suitable to answer the questions that we have?
Yeah, I think even within the context of a traditionally conceived randomized trial, there's a lot of use of real-world data for thinking about how to structure that trial, how to define endpoints, how to think about populations where kind of efficacy can be best demonstrated, thinking about power. I mean, I think there's a lot of parts of randomized trial design that can be supported with real-world data and real-world evidence, and you can certainly take it well beyond as well to incorporate the actual real-world data, the actual real-world evidence into the trial itself. But I think even without doing that, you could certainly bolster the design of the trial through data-driven inquiries.
Yeah, so I think there are a lot of recent development in the statistics community as well as data science community in terms of methodology aspect in the design. And there are some recent ICH E9R1 that introduced the concept of estimander, which basically considering not only the treatment, right, the assignment itself is a randomized or observational, but what into current events. And so that guides your kind of endpoints definition that most reflects to your clinical questions. And so this may not, is a good concept because it's not only applied for the randomized trials, but also it's relevant for the endpoints for even integrated hybrid trials, for example. There are some new development in terms of on the hybriding side, not necessarily just on the external control. And the key questions still remain the same as what we do the randomization trial. The reason we're using randomization is to remove the confounding factors and avoid selection bias. So the same question does not, the requirement is not less, even you put, so that's why it's requiring new design and new analytical approach. And so I think that's a really exciting period of time for statisticians and data scientists to meet those challenges. And the other one that I want to mention also, it's not very simple. I mean, people expect if there's external data, it will be simple and easier to do. Maybe data is there, but it's not necessarily easier. And also, like some of the common endpoints, you want to use endpoints that most commonly collected. So you need to design your trial at the stage to say, when you experiment arm, you can get information from the real world arm, that external control data that can answer your questions. The problem here is that sometimes it's very difficult. Like survival is a very objective endpoint, but it depends on where you define your beginning time point. And in the real world data, there is no natural beginning point, right? So there are a lot of things you need to think through in the design phase when you want to use in the real world data.
Great. I love that you guys also touched on endpoints as part of the design process. I think we can certainly be selecting or be more mindful of the endpoints that we select when we do integrate real-world data and evidence into trials. And you touched briefly on external control arms. How do we really determine whether an external control arm is feasible? Can you talk me through that process?
Is that a question to me?
Could be to anyone. It's probably more towards Dorothy or Jeremy, since I think they've been, but anyone, whoever wants to respond.
I can kick it off, and then Jeremy, I think, is more the expert here. So, but for sure, I think there are some preconditions, and we should always, first of all, engage very early on with the regulators and the payers, so on the acceptance before we start thinking at all. And then it has to be somehow that you have a good understanding of the natural history of the disease, so that you can contextualize it properly. You should have, what Ying just mentioned, have an objective outcome measure like mortality, survival, and not something very vague with lots of gray zones. You should have somehow an understanding of patient comparability. So do you have the right comparisons, and do you have the right confounder information and good covariate measurements? And usually what is helpful from an epidemiological perspective that you have a large effect size, so that it is not the second dot after the very, very simple small one, but really that you have a large effect size. But Jeremy, please jump in.
Yeah, I agree with everything you said, and I think that's a pretty complete list. I just want to double click on one piece, which is the comparability. I think where external control arms have not succeeded, it's essentially it's in part in the problem area of comparability, in that what you're doing is you're creating an external control arm that's serving in place what would have been a randomized set of controls. And if you're bringing in an external control arm that doesn't truly represent what would have been that randomized control experience, then you're creating fundamentally a study that's not interpretable. And I think some external control arms fail there. So thinking very much about, is the real world external control data that I'm bringing in comparable to what I would have gotten in a randomized environment? And that's both in terms of patients and in terms of patient experience, in terms of measurement, in terms of follow-up, but also in terms of time and being able to think about the randomization process itself. And can you emulate that in some way, in terms of finding sort of a time point at which to anchor your analysis in the real world? These are all things that can affect the validity of an external control arm. They're all solvable. They just require some attention.
Yeah, maybe I want to share also some of work with ALS. Maybe so the project that funded by UCB is trying to using the concurrent registry to supplement randomized trials. So we want to test the feasibility as step one, and we look for the registry data from Netherlands, historical, maybe a trial that happened 10 years ago, and that's futile trial. And so before that, we look for the ALS clinical trials that have been published in the journal in the recent years. We reviewed 11 trials that use external controls. There are only 3 use hybrid controls. The other 8 using just external control. And only 3 of those 11 have overlap in terms of time of the external control and the trial group itself. And also usually the difference is quite astonishing, like several years historically. As the field is moving, so it's sometimes the temporal change may not comparable. And also 8 of the 11 did not even match for the eligibility criteria. And 7 of the trial did not match the sites. So there's a variety of ways to use that. We look for our Netherlands trial and data and the trial. We find, first of all, the eligibility criteria, even you matched eligibility criteria, patient enrolled in the trial still different from the population that you certify eligibility criteria. They are maybe a more severe disease and the longer term more early diagnosis than recent diagnosis. And if we don't adjust, just look for eligibility criteria, the outcome for the placebo arm and the patients are not matched. But fortunately, if you match the characteristics and the chance to be randomized using propensity score, and we find that the placebo arm can match the events overall survival with the properly controlled historical control hybrid arm. So those are the lessons we need to learn. That's really need to pay a lot of attention to the data. It's not just you take it and you can use it.
I think external control arms sort of evolved from comparator cohorts, which have been used in clinical research for a long time. And I think evolved due to a need of recruitment challenges primarily, because they tend to target, for good reason, rare populations where it's hard to find enough patients to enroll to a clinical study. So actually, one way that's been pretty interesting to work with external control arms is removing redundancy in the patient population that's actually in the control arm and also perhaps in the clinical trial. So if you start to think about, one example we worked on was non-small cell lung cancer, an ultra-rare biomarker-driven type of lung cancer. Hard to find these patients, and there was actually, what the sponsor wanted to do was pull together data. There wasn't, no one data set out there had enough patients in it, so they pulled together data from actually 3 different data sets, and it turned out there was patient redundancy across that, which is, if you're working with de-identified data as as most sponsors are when it comes to external control arms. It's actually important to know that, as even just an overlap of a couple of patients can really throw off your analyses. So we help kind of remove those, but I think that's important going forward when we start to think about rare diseases, rare tumor types. You want to avoid utilizing data on the same patient multiple times in your study analysis.
Awesome. Thank you so much for all those really rich insights. And again, examples. Examples are so helpful to put color to these questions. I wanted to move on to the next question. And really, now that we know that regulators are open to accepting evidence packages, which include real-world data in combination with prospectively collected clinical data, what are the key challenges in the context of collecting or using real-world data in addition to prospective clinical evidence generation?
Yeah, probably I think there's a lot of complementary points here to the external control arm study. So I think first of all, the mindset has to be there that real-world data studies or real-world evidence studies are not replacing RCTs, but that they complement them. And we mentioned before that you would not do a real-world evidence study if you want to show the initial efficacy of a drug, right? So I think the next step is really to identify the right use case when it makes sense to use real-world data in addition to prospectively collected randomized trial data. And then the second one is the point I made before, is really this early engagement with the stakeholders for whom you want to use the evidence you generate, be it the regulator, the payer, whoever, so that you really are in close dialogue from the early days on. And then comes again to the are the right data available, the right data sources, the right quality of the data sources, or can it be generated in a timely in a reasonable time frame? That's important. And then that you have the needed information, which we just pointed out also for the external control arm, that you have the right population information, outcome information, confounder information, so that you can really generate valid information or evidence. I think that would be for me the key points to really always keep in mind that it's complementary in this early engagement and the right data sources.
Excellent. Well, we don't have to belabor the key challenges, but I do want to kind of double click, to use Jeremy's term, on the data missingness as a key challenge. So thinking through data missingness is not a new concept. We've seen it appear in new and different ways now. So my question to you all is really, do you believe real-world data allows us to better address data missingness? And are we kind of able to learn more about it in thinking about what kind of missingness, what's causing the missingness, and what the impact of the missing data is? So does real-world data allow us to really think through those kind of sub-questions in a different way?
Yeah, I think incomplete data, it's a big barrier to adoption of the use of real-world data, probably especially in clinical development. If you think about a single source of data, such as EHR claims data, it's just not going to tell the whole patient story. And this is going to vary a lot by data type, but you're likely to be missing things like dates, diagnosis history, treatment patterns, and all sorts of important patient outcomes info. So I kind of look at this as a data fragmentation problem. So right now there's huge volume and diversity of data about a patient. And each of these data sets brings a useful element and a range of potential insights that you could draw from those data elements. But because of the way data is kind of generated in the healthcare system today, each data set probably only tells a piece of the puzzle, and it's pretty siloed. So really connecting these data together, whether it's claims data, EHR data, you name your favorite type of disparate data source, it all kind of helps tell a more complete picture of the patient and provide population outcomes to answer a variety of different use cases where the holistic view of a patient might be needed.
Yeah, I think missing data, is old and a long time problem, right? And partially statistically in the long time that if it's missing at random, we will have a way to impute. The problem here is the most data are not missing at random, and we don't know why they are missing. And I think somehow the real world data, because the volume and so may help us to understand why the data is missing and what are the contributors to the missing. missingness. And so I think that helps. Sometimes, data extrapolation will help if you have longitudinal data, which otherwise you may not be able to get from one-time cross-sectional data. And so, I think statistics can play a lot of role here. And partially, most traditional epidemiology studies dealing with the confounding, those technologies can also be used here and treating the missing data as one of the confounding factors.
And coming back to Emily's point, whether real-world data can better address missingness, it's probably it can probably better address it, but it can also maximize or increase our challenges with missingness, because if we use more patient-generated health data, we create also more sources of missingness. I think what was mentioned before, really this single source of data, this we should keep in mind, because data linkages can really help to better understand missingness and validate. So if you link patient-generated health data from sensors, for example, with EMR data, with electronic medical records, this could help us to better understand missingness. And last but not least, coming to the statistical point, I think from an epidemiological perspective, when we use transactional data like claims data, it's often, or the basic challenge for us is if something is not recorded, a comorbidity, we don't know whether it was not captured, and so it's missingness, or whether it's not present, so that it's a negative answer. So that's the challenge we have later on to evaluate with other methods.
Maybe I bring back also the previous question about challenges. It's missingness is one thing. The other one is that if not missing, the data quality may be the other question. And so very simple, like height and weight. I mean, you expect that should be very accurate. And we looked at the VA Million Veterans Project with 500,000 data point patients with about 10 million records or 25 million of the weights. And there is a substantial amount of data. If you look longitudinally to calibrate, there is maybe 15% of the data had, when you translate the quality body mass index, there 11% of the data maybe have problem in the quality, data quality issue. And its impact on the, like, say, obesity class 3, which is an important question. So without the quality control, there are 15% of veterans have that problem. If you exclude those not plausible data, it reduces to 11%. And it's better for the lab data because lab usually have better quality control. But we observe those patient report outcomes can potentially, in the transcription process, there are a lot of potential errors.
Yeah, building on all of it, I mentioned, data fragmentation and connecting data sources. And while I think that's hugely important, there are still, at the end of the day, there are some common examples where the data is just really, really hard to find. So ones that seem pretty simple. So for instance, if you're doing any sort of observational data study in lung cancer, you're going to want to know smoking status. Turns out that's really hard to find. It's almost always in the EHR, but it's rarely in a nice structured format. So, I think that goes back to not just data missingness, but data collection standards, data standardization across EHRs. And I think that plays a huge role in this too.
Absolutely. Appreciate those points. So I wanted to kind of move next to what kind of disciplines are really involved in analyzing this data. We have a data scientists on the line with Ying, and we have some epidemiologists on the line, and certainly biostatisticians are involved. So who all needs to really come together to analyze this kind of data, and how do we really think about harmonizing their perspectives to better work together?
Yeah, so I'll take that one. All of the above, right? Plus, and I think building on the last discussion, real world data are, if you go back to your slide about the sources of real world data, and then the slide about the kinds of things that we want to do with it, if you overlap the two. Very few of the data that we're working with for the purposes that we're working with them are generated for that purpose. This is secondary use of data. And that's not always the case, but it's mostly the case. And so, through the various disciplines and through the various lenses, we have to think about why were these data generated? And to the question about missingness, it's both, what is explicitly missing, like there's a field that's empty, and what's implicitly missing, like it wasn't captured, because that's not the reason that these data came together. So, to your list, I would add, people who have real expertise in why the data were captured, how the data were captured, because that tells you what to expect from the data and what to not expect from the data. And I think that's equally important. And so, people who understand the data generation process, so if it's EHR data, people from health systems who understand how EHR data in that health system is captured, what kind of patients come, whether patients, come back for every visit, or you're just seeing, the most severe cases, at the most severe moments. This is all really critical. The clinical perspective, I'd say, is also really critical. You need to understand sort of what the disease processes are that are happening, what the patterns are, and even if you get into some of the epidemiological details that I love and work with every day, confounding and instrumental variables and these things, so much of that is informed by clinical expertise. So I think all of the above and more, thinking about the informatics of it from a data science perspective, thinking about the epidemiology of it and the process and the causation from, the epidemiology perspective, that's the field in which I'm trained, how that we can actually sort of make inference from that, from the biostatistical perspective, plus everything else, I think is key to making any study work, but particular real world study given the real world challenges that we have with that.
Yeah, I couldn't agree more. I think the most important one is if you want to have impactful real-world evidence, to break down the silos, that you have really the people together who are passionate about understanding the health ecosystem, understanding the patient, putting the patient into the focus, understanding the methods, and as Jeremy said, the data generation process. And then everybody get really thrilled about their areas and bringing it all together. So I think an epidemiologist like Jeremy, myself, we get thrilled if we can design a study properly, understand how to best design it, and define the population and the comparators. The data scientists become super thrilled if they have the big heterogeneous linked data sources. The clinicians want to understand the patient. So I think to bring it all together, then you can really maximize the value of real-world data.
Yeah, I think it's no one discipline can carry out this whole task because it's really teamwork. At the end, you want general evidence. It depends on analytical methods. You want to use more robust, and you want to assess potential confounding factors. There are also for example, like causal inference can be used to help understand the counterfactor. But the difference between causal inference and exploratory analysis is that causal inference, you really explicitly specify your assumptions. Now, statistician analysts will never be able to validate or understand the plausibility of these assumptions, right? So really, the clinicians, epidemiologists, subject matter experts, really the ones that help us to understand this assumption is reasonable. I mean, you can not test those, right? And the other thing is the data source, right? The statistician get the data actually, or most of the time, has been processed by the informaticians. And there are a lot of middle steps, and nobody really the statistician really need to understand those processes in terms of whether the relationship of those endpoints, whether they are really independent, right? Sometimes the data is being massaged so many times, and they create a lot of artifacts underneath. And if you ignore it in the analysis step, you may create bias that you are not aware of. So there are so many things going on. I think it's really the fun teamwork.
Agree with all that. I'd also really double down on the importance of physicians that are trained in the disease area of interest. So right now, I think one of kind of the bleeding edge of real world data is imaging. I think there's a lot that we still can do there. And if you're, for instance, working with path reports, you should probably have a pathologist that's trained working with those data sets. The same goes, a lot of cool stuff happening and imaging for, understanding like real-world applications of RECIST criteria for tumor sizing. Again, you probably should have a medical oncologist and a radiologist involved if that's the approach that you're taking.
I love that you all mentioned that it needs to be a teamwork approach, and it's certainly we need all different perspectives represented at the table for this kind of work. It's so important. And I love that the excitement for this kind of work is really palpable. So moving from the challenges into the opportunities here, and where the excitement is, what key opportunities do you all see within real-world data and real-world evidence use within the clinical context? And do you believe that real-world data allows us to generate more representative, generalizable findings? Is that an upside? And how might real-world evidence allow us to better translate efficacy into effectiveness in routine clinical practice in the real world?
So maybe I take the first shot for the question. Basically, there are great opportunities that we have mentioned through the whole cycle of the healthcare, from the drug development all the way to post-marketing, and to even the patient level medical decision making, right? So I think the translation from efficacy to the effectiveness has been happened for in the past decades. There are a lot of efforts being done. And I always like to mention about the VA, because I was part of that in the past for 9 years, from 2009 to 7 years, from 2009 to 2006. That work in the VA Cooperative Studies Program. It is an infrastructure actually within the VA system to look for the effectiveness, so really translate the efficacy to the effectiveness. Some of the findings are very interesting because the VA CSP was the one that, was the one that actually developed a lot of cardiovascular research. And also in the mental health, that we find a lot of efficacy trial may not effective in the VA community. And those things are very important, right? So the effectiveness depends on your environment. And so I think more and more different places start to using the learning healthcare system to evaluate effectiveness and trying to move a lot of research to the point of care at the patient level, at the patient point of care. But overall, I think there is need some kind of infrastructure, both industry and academia and insurance, to really coordinate together to make this transition easier and consistent. And so I think these are great opportunity to collaborate, both academia and the industry, and also the high-tech community.
My head goes to thinking about two separate related things. One is sponsor burden, the other is patient burden. So I'll start with sponsor burden. FDA has put out some really meaningful levers to get drugs to market faster, with things like breakthrough therapy designations, accelerated approvals, and they seem to work, but they come with a lot of burden in post-marketing studies and understanding, okay, what are longer-term impacts of these new drugs that are approved, and they were approved on phase 2 studies in 100 patients. So there's actually quite a bit of burden to really follow through on the sponsor side to conduct successful post-marketing studies, to conduct successful phase 3, and really demonstrate that these products are making a meaningful difference in patients' lives. I think real-world data can really help there, especially if these drugs become commercially available. You immediately you set up things like pragmatic studies, registries, and you start to collect this information on patients over time to really get a sense of how these drugs are performing. On the side of patient burden, some of the more innovative things happening in R&D right now are around cell and gene therapies. Those come with some pretty big requirements to continue to follow patients. I think it's 15 years is kind of the standard number that we hear. 15 years of data collection on a patient, that can get kind of invasive, and you can imagine that patients are lost to follow up because it's just overwhelmingly burdensome. So if you can start to figure out better ways to actually track outcomes in these patient groups over the course of 15 plus years and utilize real world data instead of requiring that they continue to interact with a site or an investigator I think you can really reduce patient burden while still getting meaningful understanding of how these new cell and gene therapies perform in patients.
I go back to this notion, and this was something that got brought up in a forum that I was a part of maybe two years ago, of there not being there not being a gold standard, a single gold standard. RCTs are often held up as the gold standard, but maybe there's multiple gold standards out there, and efficacy translating into effectiveness. Maybe these are actually two very distinct concepts, and we certainly need to understand how a drug performs in a controlled clinical environment, and I think RCTs tell us a lot about that. But in terms of your question about generalizability and representativeness, it could very well be that those findings don't translate one-to-one into the real world because of real world things that happen outside of that controlled clinical environment. And so I think real world data absolutely give us the opportunity to really fully explore how a drug works in kind of a real patient environment, which includes all the different kinds of patients, all the different circumstances, all the different cases that happen in the real world that you would, by design, control for in a randomized trial, because you're looking to get a baseline level of understanding. So I think it's really helpful to think of multiple gold standards and multiple things that we need to understand about a drug, and think about the best way to understand each of those. And some of that is going to be randomized trial data, but real world data, real world evidence, I think, has a real opportunity to help paint a fuller picture of drugs' benefit in all the populations in which they're used.
I really like the example of several gold standards, and for me it depends really on the research question. If we want to understand the adherence from patients, then probably in a randomized controlled trial and under very strict conditions is not the best way to do it. Whereas if we want to understand the effectiveness of a drug in real world data, and we understand that there are two drugs which have the same indications but are very differently prescribed to patients depending on their risk profiles, then probably a real world evidence study does not work. So we need a randomization. So it really depends on the question. So really appreciate this approach of thinking of multiple gold standards. Thank you, Jeremy.
I'm going to steal that, Jeremy. I love that.
So maybe I want to comment also. The observational studies is great, but if possible, you can still introduce randomization into the real world data. The VA has this point care randomization trial that the equivalent drugs looking for effectiveness. If the patient and doctor have equipoise, then you can randomize, let computer decide which one to use, and you monitor the long-term endpoints through medical records. And one nice part of that is through Bayesian re-randomization, you integrate implementation of effectiveness with the exploration of the trial. So I think there are multiple innovative ways that you can do into the real world data.
Yeah, and I think that's why this approach to continue to consider real world evidence as a continuum, where it is probably very low, the real world evidence in a clinical trial, and then in a pragmatic or a simple trial, it increases, and in a completely non-interventional study, it's going to the maximum. So this continuum of real world evidence generation is for me, always a good picture.
And I'll just bring up a point that somebody else made in a public forum a few weeks back, which is that, I firmly believe this as well. It's not real world data versus randomized trials. I mean, this is not a either or. I think you just gave the example of how these really come together. They're different techniques for different purposes, and you have to think about what you're trying to study, what you're trying to understand, and think about the right technique for that. And not only is it not one versus the other, it's not even a dichotomy. We've talked about external control arms. We talked about, a point of care randomization. Like these really come together as concepts. And so, like Dorothy just said, it really is a true continuum, and we have to think of it as such.
I love that. Yeah, definitely. It could be an and it doesn't have to be an or. You can do both kinds of studies, and they're all fit for purpose for the population. So moving us along, I realize we have 4 minutes left, so I'm going to try to accelerate a little bit. What gets you guys most excited about the use and the value of real-world data in randomized controlled trials? From a pre-market perspective, can real-world data actually bring us closer to time and cost savings in completing clinical trials? And from a post-market perspective, could real-world evidence help us to better predict outcomes of interest and ultimately bring us closer towards value-based healthcare?
I'll just give a quick answer to this, which is, yes to all of the above. And I think, over the last hour, almost an hour, as you pointed out, we've explored all the different ways in which real-world evidence can complement randomized trials and even, help kind of direct the design and implementation of those trials as well. So I think the short answer to your question is yes. Much that we can do today and much that we can kind of hypothesize about for opportunities in the near future, given the wider availability of data, like you showed in your original slides, like given the abilities to link data sets together, like Devin was talking about. I think there's some great possibilities coming up as well.
Excellent. In the last minute or so, a couple of minutes, I wanted to really dig into what's possible now but isn't happening. And if you had a crystal ball, what do you believe is possible to realize in the next 5 years? And I'd like to do a quick round robin. But yeah, just really thinking through this emerging reality and this renaissance that's happened in this field. What's your vision for a paradigm shift going forward?
I think those 116 studies or regulatory submissions that included real-world data as part of the submission, I think we'll see a real shift in those types of studies actually incorporating real-world data, as not just secondary to here's our clinical trial data set and analysis. I think the real-world data will actually be included as part of that. So that's something I'm excited about and looking forward to.
So I think that the burden for the investigators will reduce if the integration between the clinical trial data capture and the real-world data system. So that will allow the, investigator clinicians and the community to engage in clinical trials. And I think that's important not only for the physician, but also to the population that engage into the trials. So you provide opportunities, not just for the patient academic center.
Maybe I'll go, and Dorothy, you can have the last word. I think there's a lot, and you mentioned sort of the axes of new data, better data quality. We have new study designs, new endpoints in terms of what we're truly trying to understand. I think there's developments in each of those directions. I'm really excited about sort of the continuous generation of data and the continuous availability of data, and I think that gives us a much longer time horizon to think about with much richer data at each and every point. And I think we have to think very carefully about how to use those data in a context, especially if we're going to use that in a regulatory submission or to support clinical guidelines or whatever the case might be. We have to be very thoughtful epidemiologically about how to use that data. But I think there's some amazing possibilities that open up as new data become available and as our acceptance of the use of these data in a continuous manner versus a point-in-time collection process that's followed by a single analysis becomes much more mainstream, widespread. Maybe that's the right word.
I think I cannot add a lot here and couldn't phrase it better. So probably just my last word here is really the patient focus as well. So how can we engage more with the patient, that the patient decides with whom they share their data so that the linkages becomes easier and that the patients are directly incentivized for sharing their data and getting insights. Thank you very much.
Awesome. I couldn't have ended it better myself. Thank you so much for all of your participation today. I think, really the key take-home message for at least me is that, research and the data we use within it should be more reflective of the real-world settings in which our patients live. And so by implementing real-world data and evidence pre-market, we can hope to close better gaps in care and improve patient outcomes in the real world and post-market. So thank you all for your participation today. Really appreciate it. And it was a really robust discussion, and have a great rest of your day.
Thank you.
Very much.
Yeah.
Goodbye.
Bye.
Bye.
Everyone.
Bye.